Everyone, very excited for our second video here. This one, we want to dive a little bit more into some of the work we do around pathogen reporting and identifying different pathogens in the barn. Here today with CSO and co-founder Dr. Jake Byrnes. Jake, do you want to introduce yourself super quick and maybe
Yeah.
give the 15-second overview of what we do, and then can ask you some sort of frequently asked questions we get.
Yeah, thanks, Michael. As Michael said, I'm Jake Byrnes. I'm the CSO here at Barnwell Bio. My background is in population and evolutionary genetics and genomics. I did my PhD a million years ago at this point. Did a stint in direct-to-consumer genetics and genomics with Ancestry, the genetics genealogy company, and then did some human pharma work, and then actually worked on human wastewater. It's where I met Michael and our other co-founder, Casey. And then made my way to working on Barnwell with these guys. And very briefly, what we're doing at Barnwell is we're building on this idea that we worked on with human wastewater, where we can collect a environmental specimen or environmental sample, which represents a whole barn, and then apply comprehensive testing. And we use metagenomic sequencing. And the idea there is we can detect anything that's left DNA in the barn. And we can do this week after week. So we get proactive biosurveillance in place and we can detect pathogens, hopefully before you even see any clinical signs. And the idea is earlier detection leads to earlier intervention, leads to better outcomes.
Awesome. Well, let's get into some questions. I guess the first question is, what kinds of pathogens can you all detect and can we detect?
Yeah.
And what level of detail can we get?
Yeah, as I said, typically most diagnostics out there are designed for individual or single targets. One of the powers of shotgun metagenomic sequencing is, I often describe it as instead of fishing with specific bait and trying to pull a particular fish, you're actually dropping that trawler net and really kind of taking a sample of all the DNA that's floating around in a barn. And that means we can detect bacteria, viruses, fungi, even eukaryotes like roundworms. So you can really sort of detect anything that's running around in your barn. And then in terms of level of detail, it's really interesting. We sequence quite deeply. We use 50 million reads of short read sequencing per sample. And because we sequence deeply, we tend to get a lot of data on each target. And the more data we get, the more specific we can get in terms of detection. So we can almost always get down to species and frequently can get all the way down to strain, which for some things is really important. As an example, for salmonella, you really need to get all the way down to which serovar is in your barn. Some aren't a problem, some are a really big problem. So you need that level of specificity.
Yeah. One question we often get is, producers will have one specific pathogen issue. I guess in your view, what's the benefit of our approach versus some of the traditional diagnostics that are out there when that's the case?
Yeah, I think an important point to cover there is typically that mindset I think comes from uh, you know, a lot of confidence around the specific target that you think you're dealing with. But often there's a lot more than, a single pathogen leading to a single problem in your barn. There's lots of documented cases around respiratory syndromes where it's really kind of the interplay of lots of different pathogens and sometimes even commensals that are typically beneficial that are actually playing a role too. So, I would say you get a lot more holistic view of what's happening in your barn with our tool than you would get for single diagnostics where, you're literally going after one particular thing. And if that thing doesn't pop, you're kind of left with the big question of where to go next. Where with our tool, you're getting that kind of full comprehensive view, and it's going to give you probably lots of clues and ideas of what's causing your problems. I think the other important aspect or dimension of what we're looking at is as you start to deploy interventions, those interventions are generally meant to be targeted for a particular pathogen, but that might not be the only thing they affect. And so one of the other things you get by deploying our tool or our platform is you can sort of monitor holistically when you apply an intervention strategy, what is it doing to the entire barn microbiome?
Um, I think another piece too, maybe you could speak to quickly is, the fact that we are collecting a sample from the whole barn versus individual animals.
Yeah.
Even if you do have a sense of kind of what you're looking for, can you speak to potentially why the sort of aggregate approach might be better?
Yeah, yeah. Importantly, we have this sort of aggregate on the side of targets, which is what I already spoke to. But there's also aggregation in our sampling, which I think is what you're getting to, Michael.
Yeah, yeah.
So, right now we're using typically boot sock swabs where, any operator on farm, it's very easy to use this technology. They just put the sock on and walk through the whole barn. So what we're getting in our sample is an aggregate representation of the entire barn. And that's really important because if you have a low level infection going, some of your birds will be infected with this particular pathogen and some won't. And so the sort of traditional pattern of select individual animals and sample them, either with swabs or doing necropsy, you always run the risk of, by dumb luck, you sampled the wrong animals. And so you're not actually going to pick up the infection. Whereas with our strategy, in general, you're getting that whole barn picture. And we've seen interesting examples where we've been able to pick things up with the whole barn sample, where individual animal samples are a mixed bag. Some are infected, some aren't.
Um, so, when we generate this data across this big range of different pathogens we're looking at, from your perspective, how do we make sure that this data is actionable and something that, a
Yeah.
producer could, make a decision with?
Yeah, that's a really fantastic question. And I think that's one of the arguably biggest challenges in using this kind of metagenomic or microbiome data. Previously we've seen people try to deploy this technology where they sort of parachute in and collect an individual sample. And, every sample is going to look different. When you look at hundreds of different microbes, you're always going to see things that look a little odd. And you're sort of stuck going, what do I do with that? One of the key principles behind what Barnwell is trying to do is deploy the technology usually in sort of a continuous way. So you sample week after week in a barn. And why that makes a difference is because those regular samples are setting kind of a baseline pattern. And every barn is a little bit different in terms of what the sort of background litter microbiome looks like. And so what you want to do is you want to start to capture that background and then look for deviations. And one of the most recent features that we rolled out is alert levels, which are based on this, where we're taking our large catalog of previous samples we've collected, probably one of the largest in the world so far, and we're able to sort of baseline all of our samples against that historical data. And then we look for and we alert customers when we see a pathogen that's, two standard deviations or three standard deviations above what a typical healthy barn looks like. And that's a powerful way to say, okay, there's a lot of things going on, but the things you really care about are these things that are alerting. And those are the things that you should, take action on or at least be monitoring very closely.
Um, so we have a lot of different customer stories at this point, but is there one that sticks out to you, one you want to kind of share?
Yeah, one quick one. And it sort of covers a lot of bases, which I think is really cool. I'll try to keep it concise. We had a layer farm in the Northeast where we started doing some continuous surveillance in three of their barns. This is one of our earliest customers, actually. So, a shout out to them. And one of the things that we observed in one of their barns is they we were actually testing for fungi, and this was kind of one of the first times we were actually looking specifically for fungi, and we saw a big spike in Candida. And that's really interesting because Candida is traditionally there's a group of species that cause crop mycosis. And so what they observed is about 3 weeks after we saw this spike in their data, they started seeing symptoms and signs of crop mycosis in some of their birds. And traditionally, this is a no antibiotics farm. So traditionally, what they deploy is copper sulfate in high dose as a litter amendment, and it's known to be kind of a broad spectrum antimicrobial that knocks down pathogens. And their traditional treatment was a pretty heavy dose of this stuff, and they apply it for up to 3 weeks. And what was interesting is first we had this detection of the candida indicating this infection, but then we followed up and continued continued monitoring during the application of the copper sulfate. And one of the things that, one of the outcomes of that analysis was seeing a really big shift in the whole microbiome. So all the commensal things that were in the barn were actually changing pretty dramatically. And a lot of bacteria that are generally associated with better production outcomes started to decrease during that copper sulfate treatment. And a lot of things that are traditionally associated with lower production outcomes kind of started to increase. And so the take home message for the head of operations for this customer was, oh, wow, it does look like, this is knocking down candida, but it might be that we're kind of using a sledgehammer when we need a scalpel. And so the idea is maybe they can deploy a little less copper sulfate or run it for a little less time. And hopefully they're not kind of, injuring the overall healthy microbiome and really just taking care of the pathogen they care about.
No, that's a great example, and I think such a good example of how, we are focused and looking at pathogens, but really having that full picture can inform the overall approach to
Yeah.
disease management. Yeah.
Yeah. Yeah. Give them a
Um,
holistic data set. Yeah.
yeah. Last question. What's your
Yeah.
favorite part about working at Barnwell Bio?
Oh, easy. The people. So, and it's kind of people writ large, right? So, first of all, we've built, I think, a really fantastic team internally. Lots of motivated, great people to work with. I'm not just saying this because it's you, Michael, but it is generally true. And then beyond us, we're very new to the poultry industry. And people have been amazing. The folks, the customers that we've talked to, the academics in the space, the researchers, everyone has been sort of resoundingly welcoming and really kind of curious and interested. Lots of appetite for deploying a new technology and trying it out. And so, people all around have been really helpful for us getting going. And so, yeah, I think that's the best thing of working here is, interacting with the internal folks and the external folks alike.
Yeah, awesome. Well, Jake, thank you so much for your time. Really
Yeah.
appreciate it. If folks have questions, have follow-ups, reach out to us, hello@barnwellbio.com, or just head to our website. And look forward to more of these videos coming soon.